Thanks for the invite to come and talk to you all about the cranberry story. I got a lot. I'm going to probably move pretty quickly. I largely just want to give you a real overlay of cranberry breeding, where it's been, and where I think we're heading in the near future. There's another program at the University of Wisconsin with Juan Zalapa, and we've coordinated our efforts. So he's with the USDA out there, and we're moving forward on developing a lot of the molecular aspects of understanding the genome and of cranberry. I'm not going to really touch too much upon the molecular aspects of it now. I just want to give you an overview of the phenotypes that we have out there, and hopefully we can enhance the cranberry for today's current climate, if you will, and needs. Cranberry is a member of the Ericaceae family. Just want you to point out, you've got two sides of the family. You've got the Vaccinium, which is the blueberries and the cranberries. And then you have the dry dehiscent side of the family, the rhododendroideae, but the one thing they have in common, you have some tropical ones like the Cavendishias that are also in the Ericaceae, but the one thing that they all require are acid soils. And our typical soils in New Jersey for cranberries and blueberries are around between 3.5, 4 pH. The other thing that's common to this family is that pollen meiosis leads to four products of meiosis like in most plants, but they're held together, and I won't get into it today, but we actually can do tetrad analysis with these pollen grains. We did some work with Rodomiro Ortiz, who's also a Wisconsin grad, on the chiasma formation between the centromere and the translocation. As I mentioned, that these are both these both fruit crops were domesticated on the East Coast. Blueberries in New Jersey, it's an autotetraploid with 48 chromosomes. The basic set is 12. American cranberry is a diploid, thank God. Makes life easier with 24 chromosomes. And what I want to start this seminar out with is that the contrast between blueberry and cranberry, but it could be cranberry and apple, it could be cranberry and peach. I think cranberry essentially decided, what's the function of fruit really? It's the dispersal of the seed to procreate the species. And blueberry, I think it's quite obvious because, we used to do the job, but now bears and birds do the job. But cranberry, I think if you look at the fruit chemistry, it really diverged to just another mode of seed dispersal. And everywhere you go with cranberry, water is usually involved. And just to contrast the two, cranberries, as you saw in the previous two slides back, is very buoyant. Brix is a little bit misleading because Brix that's essentially a measure of soluble solids. In most crops like Bruce's grapes, that's mostly sugars, I believe, and in peaches. But there's other things that are soluble, such as organic acids. And cranberry typically has a Brix of about 7 to 10%, but the profile there is quite different than blueberry. If you look at the quantity of glucose and fructose in blueberry, you're talking about 7 to 10%. In cranberry, it's 2 to 7%. I gave you the range, but most of the cranberries that you see on the market that are produced commercially are probably in the order of about 3%. And what everybody's familiar with when you eat blueberries is that linalool, which is the principal volatile that makes it taste like a blueberry. And in cranberry, it's 1-alpha terpineol, and if you look in the Merck index, you're going to see antiseptic as a description. So, and that's where probably some of the folklore has come from, the standpoint of being medicinal, which I'll get into a little bit later. The other big thing is that titratable acidity with blueberries is about 0.5 citric acid equivalents, whereas with cranberry, it's 2.5. That's 5 times more acid than cranberry. And as I'll get into maybe later, what we perceive in a fruit juice is a balance between acids and sugars. And the one issue with cranberries is relieving, a lot of the products you may have heard are receiving a lot of criticisms because you have to add quite a bit of sugar to it to make it consumable. The other interesting thing is that cranberry produces about 1/10 of 1% benzoic acid, which virtually no other, as I understand, the crops do. There's one Arctic bramble that will produce this, but benzoic acid essentially prevents fermentation, period. There's some wines, I don't know why anyone would drink a cranberry wine, but there's a winery that produces cranberry wine in New Jersey, and they want green cranberries because benzoic acid doesn't start to be formed until ripening. And if you use ripe cranberries, the benzoic acid essentially kills the fermentation. If you look at a lot of your drinks, they'll put sodium benzoate in there. Everybody thinks it's a nasty preservative, but cranberries produce it naturally. The other thing is that cranberries produce very high levels of proanthocyanidins compared to other crops. And proanthocyanidins are tannins, condensed tannins, that basically are there to tell you not to eat meat. If you bite into a green banana, those are the tannins of basically giving you that astringent mouthfeel, telling you essentially it's not ripe yet, it's not ready to be eaten. Well, cranberries, even in what we call the ripe fruit, produce proanthocyanidins. Cranberry is a diploid, as I mentioned. Here's a cell in diakinesis showing ring bivalence, rod bivalence. And if everything goes normal, you get separation. And the genome is about 570 megabases. So it's a it's not a huge genome, but it's not the smallest either. This is what the cranberry flower looks like when it first opens. You'll notice that you got they're hermaphroditic. You have a whorl of anthers, and these anthers will release their pollen with the bee visitation. After 3 days, you have a style growing through the whorl and becoming receptive. So there's a mechanism to try to at least encourage cross-pollination. But they're fairly self-fertile. We've got, actually, this is two lines, a variety called Pilgrim I started with about 20 years ago. Somebody was asking me, what's a generation? Well, I started this selfing back in 1988. And so we're only up to the S9 now. But we're sequencing this line here, Ben Lear. We have an S9 of it. And so hopefully that will make things a lot easier to assemble at the end. Just to give you a little bit of history, the industry was started off with native cultivars. Early black was selected out of Massachusetts in 1852, Howes in 1843. Early black is still being grown today in Massachusetts and New Jersey, but they're slowly being replaced by higher yielding varieties that we've released recently. You got natives from Le Munyon, which actually gave rise to one of the varieties that I just recently released. Ben Lear is still a stable, and that was selected in 1907. What cranberry is most noted for is the flavonoids. I think there was some work done here by Dr. Sung on the antioxidant capacity in cranberries based on this chemical reagent test that show the highest antioxidant, and it's largely because of the flavonoids, and that includes the anthocyanins, the red pigment, the flavanols. Cranberries have a high level of flavanols compared to most, virtually all of the fruit crops. And then, of course, the proanthocyanidins, but they all come off the shikimic acid pathway. Just to give you what proportions of various compounds we have in cranberry that I brought with me, you can see that there's two bars here to represent the germplasm variation, but you can see by and large the proanthocyanidins, based on a weight basis, provide the most amount of flavonoids. The anthocyanins, the red pigment, which occur largely in the skin, is here, and the flavonoids are fairly high. Resveratrol has been found in cranberries, but I think it's quite small. I don't know what it is in grape. I can't tell. Okay. And then triterpenoids have been some work done with anti-cancer work, but they occur at very low combination. But anyway, where all the scuttlebutt was 20 years ago was that certain compounds in cranberries prevented E. coli, which is the main culprit in pyelonephritis and cystitis and urinary tract infections, prevented the ability of the E. coli cell to adhere to uropathogenic, to cell surfaces in the bladder and in the kidney. And it was largely, what we found was that these proanthocyanidins, which are polymers of epicatechin and catechin, in grape and in cocoa, they occur largely as linked units with a single bond. Cranberry has the unique property with two of the units here having a second linkage here, ester linkage between these two units. And what that does, it fixes the rotation, or it does not allow the rotation here, and it interferes with the E. coli in docking to certain cell surfaces. Now, I have to point out that this is in a test tube. Showing it in vivo is another story. Just to give you a little history of where we've come from breeding-wise, the first breeding and selection cycle was initiated in 1929 by the USDA, and New Jersey was chosen for the nursery study because there's a disease called false blossom, a phytoplasma that devastated the industry, reduced the acreage in New Jersey from 13,000 acres to 2,500, and we're only come back about 3,700 acres now. But that's where the first breeding and selection cycle occurred, and they wanted to develop varieties that were resistant to the false blossom, and they really didn't develop any that were resistant, but they improved upon some of the qualities, the yield components and the color and that sort of thing. And from that program, there were 6 varieties released: Stevens, Pilgrim, Bergman, Beckwith, Wilcox, and Franklin. Stevens is the most widely grown cranberry variety to date. You'll find it from Chile all the way through Canada and in the States, and it's still, I would say, probably comprises over 50% of the acreage. There were 40 selections that were additionally made, and the 6 varieties were released. And it was all published in Cranberries magazine in 1947. There were about 30-some crosses made, and you can see that the Stevens and Pilgrim came from just 2 crosses here. And then this one here, I just want to point out, was never named number 35 of the 40. But it's one I found in my breeding program to be a very good parent for yield and fruit set and just general vigor. So in the 1980s and 1990s, the program that I started, we were largely after yield, color, early season ripening, and bed establishment. Disease resistance was always an issue. We're planting them out in the field, so they were always subjected to to disease pressures. But we had enough chemistries basically to control the insects and disease. One thing I want to point out is cranberries, as opposed to apples or other crop species, produces both vegetatively and sexually through stolons. The stolons are, the vigor is used to colonize, to establish a new bed, and these will root in a matter of a couple of weeks. It's amazing how quickly, you don't need any hormone or anything. Now, what I want to just show you here, there's an issue with nitrogen because this is an old slide. I apologize for the quality, but there's 10 cultivars where we did a nitrogen experiment with Joan Davenport with no, medium, and high nitrogen. And what I want to show to you is that when you start increasing the nitrogen, you notice that yield goes down in virtually all the cultivars. Most severely, more severely, like with cropper, it almost goes down to nothing. Number 35, as you can see, is a much more stable under higher nitrogen in crop production. You still, the dashed line here represents biomass, stolen biomass. But what I want to just point out to you is that the response to nitrogen has a large genetic component. The other problem with cranberry, as we were talking at lunch today, is biennial bearing. This year's crop that's going to be produced this summer, at the end of the summer, the plant has to make a decision whether to put on flower buds at the tips of the vertical shoots for the next year's crop. And some varieties, when there's an excessively, this is over like I think 10 years, excessively large crop, the bud set is lower the following year, and the crop drops. So one of the issues is biennial bearing, and this is not uncommon with a lot of woody perennials that have to do that. Hopefully everything goes right, and this is a variety Mollica Queen showing a fruit set that's actually, most of the uprights produce 4 to 5 flowers. And if you look in the old literature, you're expecting to get 1, maybe 2 berries per upright. Well, the new cultivars right now, one thing I think we've genetically enhanced is the ability to set more fruit on an upright and carry it. These are what the plots look like when we started. This is in New Jersey. This is in Wisconsin. This is, it's not in July, Mark. There's a little bit of snow left, but they're still pretty well covered. But this is what our plots look like, and how they're maintained. And if all goes right, you release a variety, Crimson Queen. This is a variety that the average in Wisconsin production is about 28,000 pounds per acre. This bed a couple of years back came in at about 62,000 pounds per acre. And one of the cultivars we released this year came in over 740 or 74,000 pounds. So I think yield-wise, we've done quite a bit of a gain. And you have to remember is that your inputs are pretty much all the same. Your fungicide, you're not spraying any more fungicides or insecticides, or maybe a little bit higher nutrition to carry the crop. But essentially, you've increased your return on your investment. And 2 varieties that are early that we released was Crimson Queen and Demoranville. And these were selected for both higher color, earlier ripening and yield. And you can select in both directions. I don't know if everybody had white cranberry juice that Ocean Spray produces. You know what the big sale was for that? It was for these McMansions in New Jersey that had white carpeting with young families. So when they spilled it, they wouldn't stain the white carpet. But needless to say, the product is not doing well anymore, so nobody is growing this variety. This is this variety here essentially quantitatively just does not produce any color. The one berries that I bought are Crimson Queen. You can see that are very highly colored and might have a useful market in the fresh fruit. You can see what the time is. Okay, so those were the the objectives up to about the year 2000. But as I mentioned, New Jersey has a pretty heavy disease pressure, particularly fungal rots. And this year, one of the main fungicides that is being used to control fruit rot, not just in New Jersey, Massachusetts, Wisconsin, and the Pacific Northwest, is chlorothalonil. The trade name is Bravo. And the European Union has just reduced the MRL to essentially 0. So you can't use chlorothalonil to control fruit rot in New Jersey, and if you're going to ship to Europe. On top of that, California's got Proposition 65, and chlorothalonil is listed on that list as a nasty substance. And there's also another problem is that chlorothalonil has been linked to colony collapse disorder, possibly. Well, like somebody's mentioned, everything's linked to CCD. But it's obviously on the radar screen. So my program right now is largely focused on fruit rot resistance. A lot of you are eating sweet and dried cranberries. Well, up until Ocean Spray developed those about, I think it's about 10 years now, maybe 15, the processing market did not have a lot of requirements as far as berry shape, size, or appearance. This SDCs, as they call them, they prefer a certain color, not too much, not too little, a round shape, large fruit size, good fruit firmness, and they want a ripening early and late in the season. Just to move into the bulk of my talk right now is germplasm, because that's basically what fuels provides you the genes to modify your cultivars. Macrocarpon is native to northeast U.S., largely east of the Mississippi and southeast Canada. There's another species, Oxycoccus, that's tetraploid that occurs across Canada. It's into New Jersey, and it's actually here, it's in New York, in the Adirondacks, and also in the Appalachians down here. There's a diploid species up in Alaska that we found that I'll hopefully talk about a little bit later. But you'll find it along water. This is a lake right down from the station. You're all welcome to stop by in the middle of the Pine Barrens, but you can see cranberries on the side here. And this is one of the pH of this water is about 4. You'll also find it in the dunes in Cape Cod. And if you look in these hollows down here, you'll find cranberry populations growing in those areas as well. And you'll also find them along river banks through Pennsylvania and New York. And somebody was mentioning here that they were collecting cranberries around here. I'm assuming it was probably around the water somewhere. Anyway, I went out and collected both wild accessions from the wild, being a native to North America, and I grew them out in these plots. So this is essentially my germplasm, one of my germplasm beds. And I just wanted to point out is that you have variation for, if you look in the literature, all cranberries produce stolons. Well, there's a very tiny amount that do not. They, obviously from the standpoint of usefulness, they're probably of little value, but you do find variation for how it partitions its vegetative resources. Now, this germplasm collection that I assembled, I was working with Ocean Spray at the time, and they had a nice HPLC group. And so we subjected the fruit of all these germplasm accessions and just wanted to get an idea what the variation was for the fruit constituents. Proanthocyanidins aren't on here. We used another test for that, and I'm probably not going to talk too much about it. But the anthocyanins is what's the principal quality feature that Ocean Spray was and it still is interested in. And the anthocyanins in cranberry are basically 6. There's cyanidin galactoside, cyanidin glucoside, very little of that, and cyanidin rabinoside. The cyanidin gets methoxylated to pianidin, but it's largely, they occur as galactosides and arabinosides in the American cranberry. So what I'm going to do now is I'm going to just take you through some of the traits in that we're working with in this germplasm collection to hopefully improve cranberry. As I mentioned, we got a problem with cranberry fruit rot. If we don't spray fungicides at all, we'll essentially get 100% fruit rot. So from the breeding standpoint, it's great because you got the selection pressure to do what you need to do. For the grower, it's not so great. The MRLs and the Prop 65 in California is what's driving this. Somebody mentioned what pathogen. Well, there's over 15. There's 5 principal ones with Phylosticta, Vaccinium, Phycolospora. They've got the pathologists or the taxonomists keep changing the name, so I can't keep track of it. But 1 year, one will predominate, and another year, another one will predominate. But this is all, and all the infection occurs largely during bloom and early fruit set. So what we did is we took our, I guess it was about 400 or 500 accessions, and we essentially stopped spraying fungicides on this germplasm bed that I showed you. And you can see that we rated rots up to 5. This is essentially 100% fruit rot over 2 years. And if we didn't have any of these little bars here, I probably would have wrapped it up because the genetic variation wasn't there. But luckily, we think we found a few that have some level of resistance. And so we did the typical SCARs and the SSRs, and we compared whether they were at all related. As it turns out, they seem to be 4 different, what we call types, and they are no more related to one another than any other cranberry. So I think we've got some sources of resistance that hopefully maybe where the resistance is a little bit different. So the ultimate goal here, at least in my program, is to try to combine the resistance alleles from the various sources into one genotype and hopefully enhance resistance more. We made a whole slew of crosses of of these with our most productive types like Crimson Queen and Malacca Queen that I showed you. And these are just frequency distributions of the resistance where if you see the green and the blue here, those indicate there's some level of resistance. So if you cross resistant by resistant, you recover a lot more resistant types than if you cross two susceptible ones where you don't see any blue or green here. So essentially, there is a genetic component to the resistance. And just to show you what we grow them in, these are 2 5-acre beds with 3,000 plots each. And if you go down, so this is what our current breeding populations look like. And as I was mentioning to somebody, how long does it take you to get there? Well, it's at least 3 to 5 years until you can start actually even evaluating. So it's a very long process. We did a parent offspring regression on these, and essentially at least some of the resistance seems to be heritable in an additive way. The dotted line here represents what you would expect. So most of the resistant ones are a little bit more fruit rot susceptible than what would be predicted on the mid-parent value. So there appears to be some sort of recessive nature to at least to the resistance in general. So we want to take these into the second and third breeding cycle selections and intercrossing some of the selections out of here. So I think there's hope for at least enhancing some level of resistance. Immunity is probably not possible at this point. But just to point out, Malacca Queen and Stevens, these are separated into sound fruit, somewhere in the middle, and rotten fruit. So this shows you the yields and the size. We have 2 selections from Bud's Blues, one of the resistance types crossed with Malacca Queen and Crimson Queen. This is the sound fruit, and this is the rotten fruit here. So you can see this is a replicated trial with I don't think we completely eliminated fungicides, but we were down to about, I think, 1 spray. Normally we put 5, 6 sprays on. So I think we've made some genetic gain for resistance and yield, so I think there's hope. I'm just going to take you through a whole series of things that we found in the germplasm that are Mendelian. Just give you a little sense of what sort of types we have. We have what we call Murphy's green. Anthocyanins in the leaves basically are UV protectant for chlorophyll. And there was one clone they called Murphy's green from Massachusetts, and this segregates in a recessive manner in a single locus model. What's interesting is that the flower, the pedicel, and the base of the anthers is also doesn't produce anthocyanins, where a normal flower does, but yet the fruit expresses the anthocyanin. So it's probably some regulatory allele that's not functioning in the vegetative portion. There are also yellow cranberries, and this was found in a germplasm in Maine, and this segregates in the 3-to-1. So we've moved it's a small berry. We moved it into larger berries, but as I mentioned earlier, the white juice doesn't seem to be doing too well. What I want to sort of touch on now is that cranberries, if you look at all the products, they're receiving a lot of heat from the standpoint of the amount of sugar you have to add. And as I mentioned, the acids in cranberry are 5 times higher than they are in blueberry. So to make something at least palatable for us, we either have to raise sugars. The other possibility is lowering acids. And we looked for variation in sugars and acids, and there is some variation for sugars, but doubling almost nothing doesn't get you too far. But I think we found some interesting things to work with when it comes to acids. You get in normal cranberry, you get about 8 to 10 milligrams per gram of citric and about 6 to 10 milligrams of malic. And we found one accession from New York that appears to have low malic. Here's a population, a frequency distribution of a segregating population. And we have it segregating where we can get the malic down to about 1 or 2 milligrams per gram. Unfortunately, there's a vegetative pleiotropic effect where I think from the standpoint of it being useful, at least in a homozygous, will not lead to anything that's going to be sufficiently productive to warrant the commercialization. This is a heterozygote that has moderately lower malic acid, and so there might be some potential there. But the normal range for cranberry is between 6 and 10 milligrams per gram. But this segregates. We're looking for SSR markers that are linked to this, so we don't have to wait till fruiting for 3 or 4 years before we can make our selection. Another accession we found from New Jersey has low citric acid, and it segregates in a Mendelian fashion. The accession is J712, and it was crossed to Stevens, and then number 35s, and then we intercrossed the two offspring hybrids from that. And this is the frequency and distribution of the segregation for that population. And we're recovering genotypes with very low citric acid. And I think if we can cut down the citric acid by 1/10, which this is roughly what it is, a lot of fruit juices are about that range. So there's the possibility of reducing the acids low enough where we don't have to add as much sugar. And what's interesting was that we were looking for SSRs that were linked to this, and we found one marker, and we found the SSR that's linked to the low acid is 217. Stevens provided the other marker in this hybrid, and number 35 provided this marker. And if you look at the distribution here of the four genotypes for the SSR, you can see that the population, there's variation for the alleles for the citric acid, because crossing it with matching it with allele 229 and 217 gives you citric acid in the mid-range, and whereas if you don't have the 217 at all, it gives you citric acid in the range that's more normal of the cranberry. So we probably wouldn't have picked this up without the SSR markers and this type of cross. So we think we have at least 2 different alleles for citric acid content in the cranberry. This particular phenotype with a very low citric acid doesn't seem to have any ill effects. What's interesting is that you notice that they should all be roughly 1-to-1-to-1 segregation. So there might be some fitness issues because we did not recover the expected number in these genotypic classes. So we're just starting to get into this, into looking at this. But I think there's at least hope for reducing acids in cranberry products and hopefully the sugars. What I'm going to touch upon now is a little bit stuff that we did earlier, but I mentioned that there was a diploid species of cranberry, Vaccinium oxycoccus. This is American cranberry. There's a couple of polyploids. These are tetraploids from, this one's from Siberia, this one's from Wisconsin, I believe. And here's the diploid from Alaska. And what's nice about it is that they produce very fertile F1s. And the hybrid is somewhat intermediate. This is the oxycoccus and the macrocarpon. And I had a slide with all the vegetative points, but everything is roughly intermediate between the 2 species. But what I want to talk about today is that the flavonoids, they're considered to be health constituents. And the one aspect of flavonoids is that they typically, many of them don't occur as aglycones. They usually have a sugar attached to it. In cranberry, I showed you that anthocyanidins are attached to either galactoside or rabinoside. Now, there's been work done that suggests that flavonoids that are conjugated to glucose are actually more bioavailable for us, and it will actually lead to higher antioxidant potential for human health. The problem is the galactoside and the ribonoside are not as bioavailable. What we found was that in the oxycoccus, the anthocyanin profile, this is HPLC for the anthocyanins, the oxycoccus profile is largely glucosides. And the macrocarpon, this is leaf anthocyanin, so the pionidin actually is not expressed in the leaf, only in the fruit. But you have the galactoside and you have the ribonoside. The hybrid is somewhere, shows you roughly 3 peaks, roughly equal quantities, but this is actually, you have 2 segregation and you recover both parental phenotypes or genotypes, or I should say phenotypes, and also the hybrid phenotype. And it segregates in a Mendelian fashion. So we can actually genetically modify the glycosylation, at least on the anthocyanins. It doesn't happen in the flavanols, which I was hoping for, but that's another story. Somebody was asking me about interspecific hybridization. This is blueberry from Florida. This is American cranberry, and you can cross it with these two and produce what's the first hybrid, I think, between blueberry and cranberry. It's got its problems, but the potential here is what we're hoping is to transfer the sugar, natural sugars into cranberry, but it's a pretty sterile beast at this point in time. But it does produce fruit, and so these are the products. So they are cousins. I was trying to cross New Jersey blueberries with New Jersey cranberries for about 20 years, never got anywhere. It took a Florida blueberry and Alaskan cranberry to get there, so who would have thought? We developed a crude genetic map a couple of years ago, and working with Juan Zalapa in Wisconsin, where, you know, obviously like the march with almost every genetics program, I think today, is getting a high density map of the genome. It's been a long many years. It's probably, I hate to think how many years, but since 1988, cranberry breeding, you really have to be in it for a while, I think, to go anywhere. But it took a lot of people to support the program. So I think, well, these are I have some test trials in Oregon and in Wisconsin. And so this is where I was mentioning somebody earlier, is that it's really useful to get growers, these are all in growers' fields, is to put out some of your best material in growers' fields and have them look at it, because they see it from many cases, a much different viewpoint. And it really, I think, adds to your ability to select out a variety that would be beneficial for the industry. I think I'll leave it at that.